Okay, so this is the notes recording for chapter 24, the urinary system. Urinary system is composed of course, the kidneys, the urinary bladder, the ureters, or ureters, and the urethra. Okay. We're going to focus mainly on the kidneys. And we need to get some of the kidneys' main functions out of the way. The first main function is urine production, which is basically urine is a byproduct of blood cleaning. It also plays a critical role in homeostasis of the interstitial fluid. It does this by helping to maintain blood pressure, pH, and ion concentration. It also helps produce vitamin D3, which is called calcitriol. And then another one that we've heard of, erythropoietin or EPO. Which is used to stimulate red blood cell production. Okay. So you may have learned in lab that we have about 1 million nephrons in each kidney. And the nephron is the functional unit of the kidney. It's just a tube that is tightly associated with a blood capillary. So this is my drawing of a nephron. And I have here on the right side the kidneys that are located between the 11th and the 12th rib. And so I'm going to ask you, why is the right kidney slightly lower than the left kidney? And that's because it has to make room for the liver. Okay, so let's get some anatomy out of the way of the nephron. So if this is the glomerulus, which is the capillary system, let's back up a bit. So blood coming into the nephron, if we start off, it's coming from the renal artery, and it's going to make its way to the afferent arteriole, which has a wider diameter, going into the capillary system, which is the glomerulus, which is a wholly capillary, which we call fenestrated capillaries, and then it comes back out this way through the efferent arteriole. It has a smaller diameter than the afferent arteriole. If we continue this on, if I draw this right, we would also have a capillary or extended capillaries called the peritubular capillaries that come in through here. And it's not just a loop like this, it's actually a network or a net that surrounds the nephron. Normal ones are called peritubular capillaries. But whenever they extend even further down into the medullary region, they're called the vasorecta. And this would end up going into the renal vein. The blood would end up into the renal vein. So now what about the anatomy of the nephron itself? Well, filtrate comes from the glomerulus and ends up in the renal capsule, or it's called Bowman's capsule. Then it flows into the proximal convoluted tubule, then down the descending loop of Henle, up the ascending loop of Henle, and then to the distal convoluted tubule, and then finally it empties into the collecting duct here. So all the nephrons will be emptying into a collecting duct. the fluid will end up accumulating and then emptying into a ureter, and finally the urinary bladder. If you notice here, the way in which filtrate moves versus the way that blood moves, it is in opposite directions, and that's going to become important later on. Now let's get some mechanisms out of the way, some important mechanisms. So here we have a blood vessel with a lumen, and of course within it we have blood. Then we have the interstitial space, and then we have the lumen of the nephron tubule, and within that we have filtrate. The walls of the blood vessel are lined with simple squamous epithelium, while the walls of the nephron tubule are lined with cells like simple cuboidal epithelium, and the type of cells will actually determine the function. Sometimes they will have cilia on them, or microvilli. And each tubule is actually associated with a blood vessel. So, in essence, we're going to have 3 basic spaces, and we'll see 4 basic mechanisms. So the first mechanism is moving stuff from the blood into the lumen of the nephron tubule, and we call this mechanism filtration. This is very general in whatever it is moving from the blood into the nephron tubule. So from the blood into the interstitial space and then into the lumen of the tubule. And when it is very general in nature, it is called filtration. Now, if it is in the opposite direction from the lumen of the nephron tubule, so from the filtrate to the interstitial space to the blood, we call this reabsorption. So from the filtrate back to the blood. Now, let's say we move something from the blood to the interstitial space to the nephron tubule, and we're going to say it is something that is very specific. So we're picking out something very specifically or deliberately. Then we call this secretion. All right. The last one is from the nephron tubule out, and we call this excretion. So that's eliminating filtrate, so like urination. So that's 4 basic mechanisms across 3 basic spaces. So again, just looking at those mechanisms, direction matters. So filtration only happens in this location, and that's the renal corpuscle. It's made up of the glomerulus and Bowman's capsule. Oops, sorry guys. And Bowman's capsule. It is the only place that filtration happens. The glomerulus is a fenestrated capillary, meaning it has holes that allow substances and ions and particles to move through. And on top of the capillary is a visceral layer of Bowman's capsule. It also has a parietal layer forming the outside of the capsule. In between is the capsular space that's going to hold the filtrate that is being filtered out. So on top of the capillaries are these cells called podocytes. So in this picture, I have these funky looking cells called podocytes. And they sort of interdigitate to cover the glomerular capillaries, and they form spaces allowing filtrate through. So remember, the capillary, we can see it here underneath the spaces of the podocytes, has holes within them because they're fenestrated that allows the filtrate to come through, and then it passes through the spaces between these podocytes. Okay. So if we put some numbers to this and say that 3 liters of blood, if you have 3 liters of blood, that means in 24 hours you could filter 180 liters. That means that's about 60 times your total blood volume in a day. That's 60 times your total blood volume in a day. That's quite a bit. That means your kidneys are really working. So let's talk about some pressure dynamics here. So I have here first a capillary down here. And let's review some capillary exchange. We know about hydrostatic pressure that pushes fluid out of the capillary. And let's say that hydrostatic pressure at the arterial end of the capillary is 32 millimeters of mercury. And as we move across the capillary, as blood flows this way, hydrostatic pressure lessens. And let's say it ends up being about 15 millimeters of mercury. It decreases, remember, due to friction. Now the other force is osmotic pressure. And it does not change across the capillary. And let's say it's between 25 to 30 millimeters of mercury. This is osmotic pressure. And that's due, remember, to the plasma proteins found in the plasma that pulls water back in. On average, we filter, so that means net filtration, fluid moving out, is about 3 liters a day. And that gets picked up by the lymphatic capillaries. So what does that mean for our nephron? Well, remember, the glomerulus is a set of capillaries. So of course we're going to have some hydrostatic pressure. So in our glomerulus, here's our heart pushing fluid out through hydrostatic pressure. We'll say this is 32 millimeters of mercury. And the weird thing with this is, remember that we have filtrate already in the Bowman's capsule. And hydrostatic pressure, whenever it pushes out, it hits the walls of the Bowman's capsule and the filtrate that's already in there, and it causes a push back into the capillary. So it's a back hydrostatic pressure of about 15 millimeters of mercury. So this one is back hydrostatic pressure. Okay. What about our osmotic pressure? Well, we'll say that the plasma proteins in here are pulling fluid back in at about 30 millimeters of mercury. And the last thing that is affecting pressure here is the diameter of our vessels. So the afferent arterial, we said the diameter was larger versus the efferent arterial. And that causes some pressure dynamics. So all in all, we don't have very much filtration, but due to these different diameters of vessels, we get a pressure out of about 23 millimeters of mercury, increasing our net filtration. So that means the fluid moving into Bowman's capsule. Okay. So this one is due to the difference in diameter between the afferent arterial and the efferent arterial. Okay. All right, so the net fluid filtered out into Bowman's capsule filters out at a certain rate, and that's affected by blood pressure. So your kidneys can maintain a certain amount of filtration between a blood pressure of 80 to 180. MAP, remember, stands for mean arterial pressure. So it can maintain a filtration or glomerular filtration rate, a GFR, of 180 liters per day, as long as your blood pressure stays between 80 and 180. If blood pressure drops below 80, your GFR, your glomerular filtration rate, will also drop. If blood pressure rises above 180, your GFR, your glomerular filtration rate, will rise. So there are some things that your body does to monitor this or to affect this. One of them is called the myogenic reflex. It is facilitated by smooth muscle in the afferent, that's the larger diameter, arterial. So here's your afferent arterial. And on this afferent arterial are some sensory receptors. We'll abbreviate those as sensory receptors. And these sensory receptors will respond to stretch. Okay, they respond to stretch. So let's say in this scenario, our stimulus is an increase in blood pressure. This is going to activate our sensory receptors, our stretch sensory receptors. Because we have an increase in blood pressure, and this causes calcium channels to open. Remember, calcium is important for what? Muscle contraction. So yes, this causes smooth muscle contraction. This results in what we call vasoconstriction of the vessel, of the afferent arterial, and results in a decrease in GFR, which is what we would want to maintain 180 liters a day. Okay? The second way in which your kidneys help to control the GFR is called tubulo-glomerular control. It uses your juxta-glomerular apparatus. So let's look at what the juxtaglomerular apparatus is first, where that's located. It's made up of your granular or juxtaglomerular cells on the glomerulus. So I'm going to call those JG cells. In lab, I think you called them granular cells. And also cells found on the distal convoluted tubule in the same region, called macula densa cells. Okay, so together they form the juxtaglomerular apparatus. These macula densa cells monitor the filtrate, and if they detect a change in blood pressure, they're going to send a message to the JG cells. So let's work through this scenario. So our stimulus here is not an increase in blood pressure, but a decrease in blood pressure. So the first one was an increase. This one is going to be a decrease in blood pressure. Okay, our macula densa cells sense this, and they are going to send a message to the JG cells, or granular cells. They produce an enzyme. The JG cells produce an enzyme called renin. That is going to be dumped into the blood, and it will come in contact with a protein called angiotensinogen. This is an inactive protein. Remember, this is always in the blood, is always swimming around in the blood, but is inactive because it is an ogen, which is a zymogen, and needs to be activated. So once renin turns angiotensinogen active, it will become angiotensin 1. Okay? It comes in contact with an enzyme called ACE. ACE, remember, stands for angiotensin activating enzyme. It's made by the lungs. So ACE turns angiotensin 1 into angiotensin 2. And this is the moneymaker here. It causes powerful vasoconstriction of your vessels. It will also act to increase thirst, which will increase blood volume, and in turn increase cardiac output. Ultimately, this is acting to increase blood pressure. Okay? Now let's look at the different parts of the nephron individually. Let's start with the proximal convoluted tubule. So almost everything is filtered here. In the renal corpuscle, almost everything is filtered except for red blood cells and plasma proteins. And then 70% of everything that is filtered is reabsorbed first into the interstitial space and then into the blood. Okay, so let's look at a snapshot here of a proximal convoluted tubule cell. So you see here how we have some microvilli that aims to increase surface area for reabsorption. So if this is the lumen, we have filtrate out here. Okay, we're going to need transporters to move things from the filtrate into the blood. So one we're familiar with is SIGALT, sodium glucose transporter. It moves sodium down its concentration gradient, and when it does this, it allows glucose to hitch a ride. Remember, on the other side, then, to get rid of sodium, we'll need a sodium-potassium pump. That moves sodium out while moving potassium in. What do we need for glucose? Hopefully you said a glut transporter, a glucose transporter. That will move glucose out. Water can move on through, can diffuse on through. Water, remember, likes to follow the solutes. What about potassium? There are channels that will allow potassium to flow through. Okay. The reason I'm showing you this is that we have to have transporters to move solutes out of the filtrate. And that leads us to the next thing called the transport maximum, which is the amount of particles or solutes that can be reabsorbed by the blood is affected by the number of transporters. Or in other words, the number of transporters determines how much you're actually going to be able to reabsorb into the blood. Okay? So in this example, let's use glucose. Okay? So on the horizontal axis, we have the concentration of glucose in the plasma. And then on the vertical axis, we have the rate at which it is moving. So let's do filtration rate first. With filtration rate, we have no limit. So the more you have, the more you filter. So our graph of filtration rate is going to look like this. As the concentration goes up, we filter even more. The rate of filtration goes up. What about with reabsorption? Well, it relies on transporters. Okay. It will match filtration rate till you hit what is called the transport maximum. Then no more glucose can be reabsorbed. And we just remember use glucose as an example. So let's say that our transport maximum is right here. So with reabsorption, we're reabsorbing the glucose, matching filtration rate. Oh, and then we hit the transport maximum. Are we going to reabsorb any more, even though we're still filtering glucose? No. So that means our reabsorption graph rate will plateau. With excretion, we never excrete glucose unless we've hit the transport maximum reabsorption for glucose. So it's not going to start here. It won't start until here. That's our transport maximum. So its rate will look like this. Okay? All right, so now we need to talk about the other parts of the nephron. Let's first talk about concentration. Blood concentration is typically around 300 milliosmoles. And osmolarity just really means how concentrated something is. So how many particles or solutes are located there. interstitial fluid concentration is around 300 milliosmoles. That means that together they are what is called isoosmotic. So fluid won't move between them. But at the bottom towards the medulla, it is really concentrated at about 1200 milliosmoles. That's because water continues to leave there and then gets picked up by the bloodstream. So it moves out and then moves back in the interstitial fluid into the blood and so forth and so forth. So let's take a look at each part of the nephron. We just talked about the proximal convoluted tubule. The cells there are thick. They have an increased surface area due to the microvilli. They have lots of transporters that aid in reabsorbing what we need there. So what about the descending loop of Henle? Descending loop of Henle. The cells here are permeable to water, but not to solutes. They do not have the transporters to deal with the solutes. So in the descending loop of Henle, water is leaving. So what is going to happen to the concentration? It is 300 here, and as solutes move out, as water leaves, the filtrate becomes, hopefully you said, more concentrated. So that when it gets to the bottom, it's about 1200. Now what about the ascending loop of Henle? The cells here are waterproof. They're thicker and waterproof. They're sort of like the proximal convoluted tubule. And they are actively pumping out solutes, like sodium and potassium. Basically any solute that we have a transporter for. So here we have solutes moving out. So what is that going to do to the concentration of the filtrate? It's going to make it less concentrated so that as it's moving this way, it will become less and less concentrated until we reach the distal convoluted tubule, and it becomes so unconcentrated that it can be around 100 milliosmoles. Okay, 100 milliosmoles. That means it is super dilute. That is when your urine is really almost clear. There are hormones that will allow you to continue to reabsorb water at the distal convoluted tubule and in the collecting duct that will actually concentrate the pee. We'll talk about those later. In this picture, the nephron is actually flipped. So here we have the proximal convoluted tubule. This would be the descending loop of Henle. This would be the ascending loop of Henle. And then this would be the distal convoluted tubule. So on this part, we have water leaving. And then on this part, we have solutes leaving. What's important to note here is that as it continues to move, it's establishing this medullary concentration gradient. So let's think about this. If the glomerulus is up here, this would be in the cortex. And then as we move down farther, we're in the medulla region. If it becomes more concentrated here, we could say that it is saltier as the deeper we get into the medulla. And as water leaves and solutes leave, we're establishing this concentration gradient. And these sodium-potassium pumps that are here aim to only have a difference of about 200 milliosmoles to keep this concentration gradient going. So they only have a difference of about 200 milliosmoles between the interstitial space and the filtrate concentration. There's only a difference of about 200 milliosmoles. And this keeps this concentration gradient going. Okay. So countercurrent multiplication, what does that mean? Well, we need to take a closer look at the different parts of the nephron. It's countercurrent because the ascending loop of Henle and the descending loop of Henle go in opposite directions, the filtrate. So descending goes down, ascending goes up. It's multiplication because the saltiness, the concentration of the medulla, drives water to be passively reabsorbed in the descending loop of Henle. Let me say that again. It's multiplication because the saltiness of the medulla drives water to be passively reabsorbed in the descending loop of Henle. Okay, remember what reabsorption is. Alright, so the thick ascending loop of Henle contains sodium transporters. So I've drawn the thick ascending loop of Henle sort of thicker. It uses ATP here to ship out some sodium, along with other things like potassium and maybe even chlorine. And the key thing here is that the higher the concentration of fluid or filtrate sending down into the medulla, the lower the concentration of the filtrate is going away. So let me say that again. The higher the concentration of filtrate that is being sent down into the medulla, the lower the concentration of filtrate that is going to be sent away or back up into the distal convoluted tubule. This results in the thick ascending loop of Henle creating a hypertonic interstitium. A hypertonic interstitium. So remember, hypertonic interstitium means that it is going to be concentrated. So water passively moves now from the descending loop of Henle. So we can draw that like this. Water moving out passively from the descending loop of Henle. Is water reabsorbing here? Nope. It's waterproof. Filtrate at the bottom of the descending loop of Henle will become hypertonic to the interstitium. Okay? Hypertonic to the interstitium. Now in the thin ascending loop of Henle, things like sodium and chlorine passively move down their concentration gradient. So sodium and chlorine in the thin ascending loop of Henle passively move down their concentration gradient. Okay, let me draw that differently. So what is that going to do to the concentration out here? It's going to make it become more concentrated. Okay? We won't bother with that because we just did that. Now I want to talk about the hormones that can affect the composition of filtrate. But we must first understand the receptors that will monitor them. So I have here 2 blood vessels that are associated with the hypothalamus. And on these blood vessels are certain receptors called osmoreceptors. And the name is deceiving. These osmoreceptors don't respond to osmolarity, they respond to stretch. Okay? So in this first scenario, we have an osmolarity of the blood of about 270 milliosmoles. This is slightly less than isoosmotic. And that means that this one is more concentrated. Okay? If it is a receptor, that means it has to be connected to some sort of neuron, if it is an osmoreceptor. So let's draw this neuron as it is connected to the hypothalamus. Since it is a sensory receptor and it's sending information to the hypothalamus, that would make this an afferent neuron. Okay. So according to this first scenario, how can we increase concentration? Well, we could increase solutes or particles to increase the concentration, or we could decrease the volume, right? This would be detected by these osmoreceptors. If we decrease the volume, is there stretch? No. But there is an action potential. So if there's no stretch, there is an action potential. I'll show you what I mean by that. So which of these scenarios does that match? Hopefully you said the second one. So that means that if the concentration goes up, it does send an action potential. Okay? So no stretch, you do get an action potential. With this one, there is no action potential. Because we are stretching. So it's opposite of what you would think. If these osmoreceptors are stretching, there's no action potential being fired. If these osmoreceptors do not stretch, then they do send an action potential. Alright, so what's the point of all this? Well, it helps to control certain hormones, and that's what we're going to look at next. The first one's called vasopressin, or ADH, antidiuretic hormone. It's made by the hypothalamus and stored in the posterior pituitary. The posterior pituitary dumps vasopressin into the blood, like all hormones. Okay. We have a nephron tubule here, and it would either be the collecting duct, CD, or the distal convoluted tubule. The distal convoluted tubule or the collecting duct has receptors for vasopressin on the walls of the nephron tubule. ADH would diffuse in, bind to its receptor, and this is actually going to cause exocytosis of a channel called an aquaporin to the luminal edge of the collecting duct or distal convoluted tubule. So that means within the cell is a little channel, and once the hormone binds to the receptor, it causes this channel to be exocytosed to the luminal edge. And like the name indicates, aqua is going to have something to do with water. The collecting duct or distal convoluted tubule is sitting in the medullary concentration gradient. So water gets sucked out to the interstitial space. So that's out here. So here's water in the lumen or the filtrate, and it's going to be sucked out this way. And then water will enter the blood, which travels back to the hypothalamus, and those osmoreceptors respond to stretch, and they say, hey, no more ADH. Let me say that again. So water enters into the blood, which travels back to the hypothalamus, and the osmoreceptors respond to stretch and say, hey, no more ADH. This is a negative feedback mechanism. Okay, so what does that do to the concentration in the nephron? So let's say that this is since it is the collecting duct or disconvoluted tubule, the concentration in the nephron would be about 100 milliosmoles. If we're taking out water, concentration would go up, right? What about in the blood? If we're adding water, concentration then would go back down to whatever it was. Okay. So now this is the last part, aldosterone. Aldosterone is a steroid hormone made in the adrenals, specifically the adrenal cortex. And since it is a steroid hormone, it has a cytoplasmic receptor. You remember, steroid hormones are lipid soluble, so they have no problems getting in past the plasma membrane. So the receptors can be found within the cell. Okay, so this cell right here, let's say it is either collecting duct, because these hormones act on the collecting duct or the distal convoluted tubule. And let's say aldosterone is dumped into the blood here. It diffuses in and binds to its receptor inside of the cell. Binding causes protein synthesis. This would be the lumen where filtrate is located. So binding of the receptor, the hormone binding to the receptor causes protein synthesis. We need proteins to make things. And what it makes is sodium and potassium channels. On the luminal edge. Channels are embedded on the luminal membrane of this distal convoluted tubule or collecting duct. The sodium-potassium pumps found on the interstitial edge, their activity is increased, and what it does is it starts to pump sodium. We'll do sodium first, sodium this way. But potassium this way. Potassium gets pumped into the distal convoluted or collecting duct. So one of the side effects of aldosterone is potassium secretion. It gets dumped from the blood into the filtrate. So aldosterone increases blood concentration of sodium, but decreases blood concentration of potassium. So most of the time aldosterone doesn't work alone. It works in junction with ADH or vasopressin. And this ADH, remember, acts to pull water into the blood. So if we're pumping a bunch of sodium in there, that's going to help also to pull water with the help of ADH into the blood. And all in all, what that does is increase blood volume or increase blood pressure. Okay. All right, so that's all I have for chapter 24. Happy studying.